is my distinct pleasure and privilege to introduce Dr. Melany Puglisi. Melany is a postdoctoral research scholar at the Center for Marine Biotechnology and Biomedicine. She came to Scripps with an extensive background in chemistry, chemical ecology, and natural products chemistry, a background which prepared her well for her research work here in marine chemical ecology. Melany's work here is focused on isolating chemical antimicrobial compounds from marine algae, and today she's going to speak about survival in the sea. So without further delay, Melany.
Good morning, and thank you, Cheryl, for that lovely introduction. Today I want to give you an overview of chemical ecology, and then I want to take you into sort of the new realms of where the field's going with the folks here at Scripps Institution of Oceanography. As she mentioned, I'm a postdoctoral research fellow. I'm working with Dr. Biochemical, and he's doing some very creative things in this field. Now, for many of you who've at least looked at a calendar, if not gone diving, you'll note that there's absolutely beautiful creatures in the ocean that don't have spines, and they don't have shells or carapaces, and they don't have big teeth like sharks, and they can't swim real fast. And so you might wonder how it is that these things make a living. So take, for instance, this nudibranch. A nudibranch is a shellless mollusk, and you find them crawling all over things, and they're very brightly colored. Now let's look at our nudibranch. Of course, this is my artistic rendition of the nudibranch. And imagine he's crawling across the reef or the sand flat looking for food, maybe another sponge or tunicate to live on. And all of a sudden a fish comes along, and to this fish he looks like a nice tasty morsel, lots of protein. And in the marine environment, which is very limited by nutrients, any kind of food source is an important food source. So our fish moves along, and he checks out, and he comes along, and he's noticing, and our nudibranch begins to get a bit nervous. But little does our fish know, is our nudibranch has a chemical defense. And so this we'll talk about today is biochemical warfare. Marine chemical ecology itself includes a lot of things that aren't necessarily detrimental. There's chemical cues and signaling and quorum sensing, and that's how a lot of things communicate with each other and learn how to or figure out who to mate with and things like that. But I'm not going to talk about that today. Today I just want to talk about chemical defense. What is marine chemical ecology? Well, natural products chemists have been interested in the ocean for many, many years. And what their interest was that we look for new drugs from marine invertebrates and algae, just like we looked at them from terrestrial bacteria and terrestrial plants and insects. All of these kinds of organisms make a lot of interesting molecules. And so probably around the 60s or so, people really started to look at the ocean and look for new compounds and new leads. And so during all of this time, and probably earlier, ecologists noticed when they were out watching the fact that fish don't eat nudibranchs and they don't eat sponges, and there's certain species of algae being avoided, they couldn't explain it by saying, well, you know, there's some kind of shell, there's this kind of defense, and so everybody assumed it was a chemical defense. So at some point, and especially in the early 1970s, Dr. Biochemical and Dr. John Faulkner, both members of Scripps Institution of Oceanography, made a lot of observations along with other people and said, you know, there's something going on here because invertebrates and plants make an awful lot of these compounds compared to their biomass. That's a lot of energy, so something must be happening. And so the field of marine chemical ecology emerged. And it's still a very interdisciplinary field. It involves natural products chemists, it involves ecologists, and in that sense it's a very, very well done field because you can't be an expert in everything. So you can't be an expert in NMR and designing experiments. And so that's been a great cooperation, and I've experienced that myself in our lab. And in 1996, Mark Hay published a really good paper sort of reviewing the state of marine chemical ecology. And so he defined it as an understanding of how plants, animals, and microorganisms interact, communicate, and compete via secondary metabolites. And so I'm talking about secondary metabolites today. Secondary metabolites are those molecules that an organism makes that are not used for respiration, oxidation, building cells, and keeping you alive. They have a completely different role. Now, they may keep you alive because they deter predators, they stop things from growing on you, but it's not the same thing as, say, a protein or a carbohydrate as you think of like food. Now, most of the studies of secondary metabolites in the ocean fall into 4 different categories. And the most studied is defense against consumers. And then you have antifouling, allelopathic effects, and antimicrobial agents. And today I'm going to address all of these topics at some level. And the best studied area is defense against consumers. And you can imagine why this would be. If you're scuba diving or you're snorkeling and you look out in the ocean, you can say, well, look, I just saw a school of fish swim by and they just took out this entire patch of algae, but they never even began to touch Alameda. What's going on? That gave people really a nice basis to start their research. And what we know now, and what's become very well accepted, is that most benthic marine organisms on tropical coral reefs produce secondary metabolites, and these deter fish feeding at least to some extent. Now think about it. If you look at this picture here and you see all the different organisms, you see a sponge and you see soft corals coming up and you can see algae. These things are very similar to plants because they're all very sedimentary. Very few things move around. Even a nudibranch doesn't go very far. It can't swim very fast. So it has these organisms very predisposed to producing compounds or having to have a chemical defense. And what's very interesting, and what a lot of people have discovered by studying these organisms, is that the production of these defenses can vary by geographic reason. People have tried to get to the bottom of this. Why is this happening? And some of it's happening maybe because of nutrient differences. Some of it's happening maybe because there's more predators in one area. So areas of overfishing, perhaps algae don't have to produce the compounds. And there's lots of instances where just by damaging an organism you can make them make more compounds. So it's a very intricate system and very well studied. So how do we find out? How do we test the hypothesis to know whether something's a chemical defense? Well, really the most important consumers of macroorganisms, we're talking about algae and invertebrates, are fishes and sea urchins. And so people have designed laboratory and field bioassays to test this hypothesis. And what we do, and this is a picture of a field assay we did in Guam, and in the field, and what we've done is we've incorporated our extracts or our pure compounds from a marine organism. In this case, this is part of my PhD research, so we're working on gorgonians here. And on each one of these ropes are 4 food cubes, and each one of those food cubes matches the nutrition of the source, which is the gorgonian. And then the control cubes are all nutrition, no chemistry, and the treated cubes are chemistry. And what we do is we put them side by side on the reef and we let the fish feed. And then from there we score the number of food cubes eaten. And this is an example of the kind of data you get from one of these bioassays. This sea fan is called an Allelopathy, and it's an absolutely beautiful sea fan. And it grows to be nearly 6 to 10 feet tall. And you can see that nothing grows on it. And very few organisms ever try to eat it. There's some very specialized mollusks that can live on it, and there's a couple of fishes that will eat the polyps. But if you're swimming along and you're just a random fish, you can't eat this. And one of the reasons for that is because it produces these compounds here. These are called sesquiterpenes, and most gorgonians produce sesquiterpene or terpene-like compounds for chemical defenses. And when we did the assay, what we show here is the number of cubes that were eaten off of those ropes, and this is the different compounds. So this is the crude extract here being tested against the control, and this is compound 1, or the acetate, being tested against the control, and this is compound 2, or the alcohol, being tested against the control. And you can see here these asterisks represent tell you that that's a significant difference between the two. And you see that the crude extract deterred fish feeding significantly more than our two compounds did, but at least some of that activity is due to the acetate. And these concentrations here represent natural concentrations. These are calculated as to what a fish might encounter so that we're doing something realistic. Now there's some question about origin of chemical defenses, and there's some really fantastic systems out on the reef that people got involved in and discovered, especially with things like nudibranchs, like on the first page, or sea hares. A lot of these organisms actually don't produce their own compounds. They get them from a sponge or a tunicate source or an algal source. Tuberanks can do it as well. And some of these even pass them on to their eggs. This is an egg mass of either a sea hare or a nudibranch. I'm not sure. I've got this slide actually from a chiropractor friend of mine. But, but it's a really nice slide. But what's really nice about it is, can you imagine swimming along and you're a fish and you have these eggs, and you think about eggs. Eggs are full of protein because they're just incredible nutrition source. There's a lot more protein in an egg than there is in an adult. So as a fish, this is something that you'd really want to go after. But you see here, there's sort of a sheath that holds these eggs in here, and in this sheath, the adult has incorporated chemistry to protect the eggs while they grow to a size where they will hatch and they will spread out and become adults. So this is a really fantastic system, and there's several examples of this, and it seems to be one strategy that these organisms have managed to use. Now there's also some examples where the larvae of some organisms produce chemical defenses and the adults don't. And that's a really neat system too, because you're protecting the most important thing, because as things get larger, they're harder to eat, and they save the babies. But that's a really new part of chemical ecology, and we don't know a lot about it. Another possibility for the origin of chemical defenses in a lot of marine organisms is symbionts. And so that's another thing we don't know about. But it's quite possible that for many organisms, say bryozoans or sponges, that harbor symbiotic bacteria or cyanobacteria or algae, these organisms actually make chemistry that's either transferred into the host organism or is just there, and so when something comes along and wants to eat it, they encounter the chemistry that way. So there seems to be a lot of different strategies. Now let's switch to sort of the more cryptic or unknown parts of marine chemical ecology and look at antifouling, allelopathic effects, and marine microbial chemical ecology. Antifouling is a really interesting topic because if you think about it, it's really obvious. Let's look at some of these pictures. There's absolutely nothing growing on this alga, and there's nothing growing on the sponge. And you look here and you see the anemone and the bryozoans, and there's nothing growing on these. And there's nothing growing on these anemones here. There's a barnacle settled in the middle of it, but it's not growing. So it's not actually growing on top of them. So it's very intuitive by looking at these pictures to say, hey, there's got to be something there that's stopping them from growing. Well, it's kind of a complex system. So you see the need for producing antifouling compounds because an alga requires light to grow. So if something settles on an alga and overgrows it, you end up losing light and the alga will die. You have another possibility is with filter feeders like sponges and soft corals and corals. They feed through the ocean. If something settles on here and it can no longer get nutrition, it will die. There's also just the whole possibility of being overgrown. And having something settle on you, overgrowing you, and taking all your space off the reef isn't a very good option either. So this is a really interesting phenomenon. It also has a very practical application, and so that's why it's been studied as much as it has, and that is, think about boats that come into the harbor and have barnacles on them that come from all different places, or mussels that come in and invade areas. So there's also that need to find a natural anti-fouling agent that will stop this from happening so that we don't have to put metals and tins in the ocean. And so we don't know too much about how this works. There's some cases where chemistry has been indirectly linked. There's another case, a very nice case, like Delicia pulchra, which is a red alga. And for that, they actually found that the chemistry produced by the alga induces the settlement of a specific species of marine bacteria. And that bacterium actually mediates what can settle on the surface of the alga. And that's a wonderful story, and it's taken nearly 10 years to get to, and a lot of molecular biology techniques. And that's the beginning, it's the first of its kind. So that's sort of where this field is going. People are trying to understand that. Another difficult idea to design experiments for is allelopathy. Allelopathy is looking at interactions between organisms that are trying to grow up next to an organism. So if you look at this soft coral here, you can see that around the bottom of the soft coral, nothing's growing. Nothing's growing up against it. There's some algae down here, but once you get into here, you sort of have a halo. Now, it doesn't have a problem growing up next to the other soft coral, but algae doesn't seem to grow in there. Sponges can't make a home in there. And this is what you could consider a byproduct of, or a result of, intensive competition for space on hard substrates. If you look at a coral reef, a very healthy coral reef, there's no space. Okay, you can't just come in and move in. Every single piece of the reef is growing, it's alive, and there's not a lot of space to settle and grow. So how do you stop some young upstart from settling next to you and taking over your spot? And so there's some hypothesis that you have compounds that are actually produced by the organisms that cause tissue necrosis in other organisms. Now, this has actually been documented pretty well in sponges. The chemistry itself hasn't necessarily been worked out, but there's a lot of sponges. If you take them and you put them next to each other and you allow them to grow, they'll start growing, and then you get this white line in between them because they start killing each other when they get close to each other. And there's a group in Wilmington, North Carolina that spends a lot of time working on that and is trying to work out the system. So those are the first three major topics of marine chemical ecology and chemical defenses that first had a major focus because you could see those interactions, you could see what was going on. But now let's switch to something we can't see. Let's talk about bacteria and fungi and other microorganisms that live in the sea. Infectious diseases of marine organisms are very rare. You don't have a lot of outbreaks. Sometimes you have isolated instances, but it's not something that you see very often. But in the last decade, we've had increased reports, and two of these ended up in Science. Most of them end up in sort of the major journals these days because they are so rare. And one of them is called the coralline lethal orange disease, or CLOD. And this is a very aggressive bacterium. We actually don't know if it's one species or more species, but it has been cultured maybe one time, but it wasn't kept in culture for very long. And it can spread, in a year, it spread all the way through the South Pacific through coral and red algae, wiping out entire populations. Another such event, or mass mortality, was with Caribbean sea fans. And this was a lot less specific. This was something that attacked in areas rather than just by species. And this was caused by a fungus called Aspergillus. And this fungus is actually a terrestrial bug that made its way into the ocean and managed to make a living there. And has wiped out just incredible stands of sea fans. And if you consider how long it takes a sea fan to grow to, say, this big, that's really kind of a sad event. So why does this happen? Well, people are starting to explore this, and they're starting to try to understand it. And there's ideas that maybe elevated seawater temperatures actually give bacteria, or opportunistic bacteria, and the ability to grow and compete and outcompete the other organisms that are there. There's also the possibility that just by runoff, development on islands and things like that, that you have the introduction of new organisms that can make a living. And then maybe it's just happening. Maybe we just didn't look. Maybe we didn't know what we were looking at. Maybe we have a better understanding now, and so now we're much more aware of it. But nobody's really proven any of these hypotheses. These are just ideas of what might be out there. Dr. Bill Fennicle and his colleagues several years ago started to think about sort of why there aren't any major outbreaks in the ocean. And he asked the question, are there even greater selective pressures for chemical defenses that we can't see? Now consider that 1 milliliter of seawater contains approximately 10 to the 6 microbes. That's a lot. And that means that marine algae and marine invertebrates and marine fishes and all marine organisms are constantly exposed to this incredible bacterial broth of all sorts of opportunistic organisms that could just jump up any time and take over if they wanted to. And this is a very different situation compared to, say, a forest, because although some microbes can be transferred in the air, you need breezes, you need all sorts of other things to happen. So this could be a much bigger pressure in the ocean, and this could be where the ocean is that much different in its chemical ecology. So in 1 milliliter of seawater, you're going to have things like bacteria, and you're going to have fungi, and then you're going to have other groups that are less well understood. The oomycetes and the thraustochytrids were first considered to be lower fungi or different kinds of bacteria, and they're not anymore. They're in their own kingdoms. And we're just beginning to get a handle on this because we're beginning to learn how to culture these organisms and define them and classify them using phylogenetics and molecular biology techniques. And in that mixture, you have potential pathogens, you have saprophytes. Now, a saprophyte is something that decomposes tissue, just like a fungus or like a mushroom sitting on a log. When you go for a walk, you see it breaking down tissue. That's the role of a saprophyte in the ocean. And so saprophytes are very efficient in breaking down healthy dead tissue. They can be found and cultured off of healthy tissue of algae, yet they don't break down the tissue. They don't break down the plant while it's still alive. So the hypothesis here is maybe there's some kind of chemical defense. You also have parasites, and of course there's all sorts of things that we don't understand about this system yet, that we can't make hypotheses about what they might do. But our working hypothesis right now is that the low incidence of infectious outbreaks in populations of marine organisms is due to the production of microbial chemical defenses. And it's a very broad hypothesis. And we're talking about invertebrates, and we're talking about plants mostly. We're not talking about fishes. There's been a lot of disease and that sort of thing in aquaculture that have been defined for vertebrates. But we're talking about the lower organisms that produce secondary metabolites. And to test this hypothesis with algae, we decided to put together a microbial screening panel. And it was a very difficult task because not a lot is known about what organisms really are detrimental to algae. And so we went and we worked with some colleagues, Dr. David Porter, and went through the literature and came up with several test organisms we could design assays for that would really let us get a handle on what's going on in a very quick way. And we used 2 species of marine fungi. The first is a known pathogen called Lindrothilaceae. And Lindrothilaceae is very interesting because it's one of the few species that cross-infects over different phyla. It will attack seagrasses and cause disease in seagrasses, and it will also cause disease in brown algae. And that's a very unusual thing because most of the associations that have been defined of plant pathogens are very species-specific or genus-specific. Another organism we're using is called Dendrophyella salina, and this is a saprophyte. This is a decomposer. And then we have an oomycete, which is used to be part of the lower fungi. And this is called Allelopathy spinosa. And not as much is known about this organism, but this particular group, the oomycetes, have the largest number of parasitic microorganisms to algae in that group. The fourth organism is a bacterium, and that's called Pseudoalteromonas bacteriolytica, and that's also known to cause disease. So these assays are really nice and they're very convenient, and we do them in the laboratory so we can bring our work back here to Scripps after we go on expeditions and collect things. And what we do is we take our alga and we grind it up and we extract it in solvent, and we make partitions between an organic or like a greasy part of the extract and then water. And then we test those extracts against our fungus or oomycete by incorporating our extract into the media and then pouring that into a 24-well plate. And then we take a small piece of our fungus hyphae and we put that into the well itself, and we let that grow for 5 to 7 days. Each one of the test organisms grows differently. And so here's an example of this is Lindrothilaceae. This is an example of what a result can look like. A lot of times you don't see any growth at all. But other times you see partial growth and you see morphology changes. Here's your controls. They grow completely to cover the wells, and so that's 100% growth. And then you see here that you have reduced growth here to about 30%. And even though it grows here, you see that there's these little, it looks like a flower almost. So we know that that particular extract affects the growth. And so what we like to do, our ultimate goal will be to define the chemistry underlying this process. But to start out, we had to find projects. So in order to do that, there's been several surveys conducted over the past few years. We recently did one in Guam, and then there's been several done in the Caribbean. And we collected over 120 species of algae and seagrasses from these two areas. So this is Halimeda, and this is Padina, and this is, I believe it's a Gelidia, and this is a seagrass bed here. And all of these represent organisms that we have in our survey. And we surveyed against all 4 of our pathogens or saprophytes that I showed you before. And what's really interesting is the number of species that inhibited growth to at least 80%. So these numbers mean that either the test organism didn't grow at all, or it only grew to maybe 20% of the control. So those are very, very potent extracts. And you can see here that you get anywhere from 40% to almost 80%. Inhibition of growth. So that suggests to us that we definitely picked some very ecologically relevant organisms, and that these may play very important roles in the ecology of marine algae. Not only are these active, but the potency of how active these extracts are can be incredible. And so here, this is the results of a Pseudo-Alteromonas bacteriolytica assay. This is the bacterium pathogen. And on this y-axis you see percent growth, and then on the x-axis you have the concentration that we tested it at. So 1 is the natural concentration in the alga, and we calculate that volumetrically. And all of these species here were active at the natural concentration. And right here, this black line here is our control. This is vancomycin. That's how we know our assay works. So we have positive control in this assay. And the dotted lines here are extracts that are extremely potent. This is half natural concentration. This is a quarter of natural concentration. This is an eighth. And this is active all the way down to 1/64th of natural concentration. So you can see that we have a lot of potency here. And this is a red alga called Portieria hornemanii. And what's really interesting about this particular experiment is that most of the chemistry that's been isolated previously from algae, because algae has been looked on for years and years and years, come from the lipophilic or the greasy part of the extract. It does not come from the water fractions. And so this data is all water fractions, which means that we're really likely to hit new chemistry, which is what we really like to do. And so we picked a project from the Caribbean survey called Penicillus capitatus, and this is a great little alga. It's called the mermaid shave brush, and it lives on the reef flats or in the mangroves. Sometimes you find it deeper, but mostly we found it in the mangroves while we were there. And what's interesting is that the crude extracts completely inhibited the growth of Lindrothilaceae, knocked it out 100%. So that was great. So we said, all right, we'll go with this one, because since we're working on something new, let's go with the most active. And through bioassay guided fractionation, we isolated 2 compounds, and these are called, we've called them the capisterones. And this is an NMR spectrum of the first one, which is an acetate here. And these are novel molecules. This only shows you the first one. These are both novel molecules. And the skeleton, the basic skeleton of the organism was already known. It's a triterpene or a steroid type skeleton. But this ketone here on the A ring and the sulfate on the side chain here are unusual. And then the substitution between the two molecules is here on this carbon 30. On this molecule, you have this functional group, but then on the second one, this functional group here is replaced with an OH, or an alcohol. And that would be this one here. So at this point here on the molecule, this part that's sticking up, there's one substitution. And what's really significant about that is that one minor substitution makes a very big difference in the activity of the molecule in our Linderthalassae assay. This is a growth inhibition curve of Linderthalassae by the capisteroids A and B from Penicillus. And on this axis again we have percent growth, and then we have concentration. So an LD50 is the lethal dose at 50%. So it's at 50% of the, when 50% of the organism's killed, so it only grows to be 50% compared to the control. And so that's what we defined here, and that's the number that we're interested in. And for compound A, the natural concentration is 25 micrograms per mil. And for compound B, it's 20 micrograms per mil. So at natural concentration, both of these are just as effective and very efficient, and it's a great system. But when you start to dilute these down and you get into lower concentrations, you see that the activity of B falls off fairly quickly at 0.94 micrograms per mil. That's still a good LD50, but it's not as good as compound A, which has the acetate, which goes to 0.03. And we tested these in combination, and we didn't see any additive or synergistic effects, so that makes us think that they just sort of work independently but toward the same goal. So today I tried to convince you that marine chemical ecology has been a successful marriage of chemists and ecologists. I've been working myself with another postdoc who's here today, Lick Tan, and we worked on the Capisteron project together. And I'm more of an ecologist, he's more of a chemist. And we've been able to get to the end of this project fairly quickly. And so we've, with the ecologists defining new experiments and becoming more creative with the new tools that we have, because now we have molecular biology, we have new chemistry analytical tools, we can do a lot more and a lot better experiments. Secondary metabolites play a really important role for marine organisms. I don't know that you really want to call them secondary metabolites anymore, because without these compounds, a lot of things would not survive on the reef. Fishes are very efficient feeders, and so without this, they wouldn't exist unless they grew really, really fast. And then for what we're doing now, we believe that a better understanding of marine microbial chemical ecology will lead to the discovery of new chemistry from marine plants and invertebrates. And so we're just at the beginning of this, and it seems to be promising. There were 2 other projects that came out of these surveys, and both of those led to new compounds as well. So I'd like to acknowledge a few people. The Gorgonian chemical ecology I did as part of my PhD research with Dr. Valerie Paul at the University of Guam and Dr. Mark Slattery at the University of Mississippi. And then my good friends John Starmer and Jason Biggs, who were good enough to do a lot of feeding assays and a lot of diving. And then the work we do now is microbial chemical ecology, and that's of course with Dr. Bill Fennicle and Paul Jensen. And Sarah Kelly has been really helpful with the assays. And then Dr. Lick Tan, who's been just a fantastic collaborator as far as chemistry. And of course my photos come from my chiropractor friend, Dr. Mark Chadwell, and another friend of mine who is a taxonomist, Gustav Pauli, and a very good friend, Linda Tatro in Guam. And much of this research was funded by National Science Foundation, grants to Bill Fennicle, Valerie Paul, and then some smaller grants from the Australian Museum of Natural History and the American Museum of Natural History to me. And thank you very much for your attention. Okay, the question is I indicated that there was 10 to the 6 bacteria in 1 milliliter of seawater, and where was that sample taken? That number is actually an estimate or an approximation of a lot of samples that have been taken a lot of different places. And so that's what marine microbiologists estimate as the mean number. It's not, we can't culture all of those bacteria. And people do these great experiments where they take water and then they extract the DNA out of it and they do these phylogenetic analyses. And that's how they get those kinds of numbers.
So if you just took a sample from the middle of the Pacific Ocean, you'd get a pretty good number anyway.
Yes, no matter where you go, you're going to get a huge number of bacteria. So it's a really big, it has the potential to be a very big pressure on the organisms that live there. Okay, the question is why you see the activity in the organic layer of an extract rather than water. We're not seeing that with these assays, actually. The reason people worked on the organic layers of algae and invertebrates for so many years is because it's a lot easier. Okay, we had the techniques. You can really evaporate, say, dichloromethane solvents, volatile solvents off a lot faster than you can with water. And so people just sort of didn't approach it. There weren't a lot of separation techniques. We didn't have the columns that we have now. So this is actually, working with water layers is a whole new frontier almost in all natural products chemistry at this point. So how do we do the feeding assays? Okay, so the feeding assays are really, really fast. Okay, so they're like 20 minutes. They're almost, sometimes the fish can be too fast, so you lose the assay because they eat so fast. So what we do is we take our extracts and we incorporate them into an agar. It's an agar with fish food in it. So it's all sort of absorbed. So you just soak the fish food with your compound so that the extract from the solvent comes off and the extract goes kind of in the food, and then you mix it with the agar in it. So it's usually really, really good. Early work in feeding assays, people question that, how long is it in there? And so folks would take ropes out and just sit them on the reef for an hour, hiding them from fishes, and then they would bring them back after different periods of time and they'd re-extract them and the chemistry would still be there. Even with water-soluble things, because you build sort of a matrix. But most of the feeding assays I did on the reef, 20 minutes, if that. I mean, the fish just come right in and feed straight away. They were very conditioned where we were. The question is, do we have any plans of commercializing the capisterones? And what's really, really interesting about these molecules is that they are only active against Lindorthaceae. I've screened them against Dendrophila salina. I've screened them against all of the bugs I showed you, and I screened them in cytotox assays, human bacterial pathogens, and they're very, very specific to Lindorthaceae. So actually one of the things I would like to do if I have time while I'm here is try to understand the mechanism of action behind that, because that actually might be more useful. Because we saw a tiny bit of activity against Candida albicans, which is a human fungal pathogen. But yeah, I'd like to see if there's a unique mechanism of action here, and that's something that maybe a pharmaceutical company could jump on, because they're not going to jump on the compounds right now. They're not that exciting that way. The question is, can this be a starting molecule for other things if you saw a slight bit of activity against Candida albicans? Yes, it could, except there's a lot of other things out there that are a lot more active, so a pharmaceutical company won't spend the money to develop it. It costs a tremendous amount of money to develop a drug or figure it out, and they're not going to. We have, the other part of the other 90% of our lab is dedicated to drug discovery. It's dedicated to finding new leads, and so all the other people work on projects like that. My real interest is like what's going on in the ocean and why this is happening, and so kind of I'm in that 10%. And when there is overlap, we do have a compound that came out of a brown alga called Lobafra that showed some really good potential activity in an assay that we run in collaboration with a pharmaceutical company, and they're following that up. But these, I don't think they'll follow up, unfortunately.